The overall goal of this Langmuir-Schaefer method with block copolymers is to fabricate cell sheets on various conditions of the temperature-responsive surface by controlling strength of cell adhesion and detachment.
This method can help answer key questions in the biomaterials field, such as biocompatible materials, cell culture surface, and bioseparation. The main advantage of this technique is that the interaction between temperature-responsive surface and cells can be controlled. Hence, the modifying conditions for cell sheet recovery can be optimized.
Though this method can provide insight into interaction between cell and surface, it can also be applied to other systems such as bioseparation and biofilm. 初めてこの方法に取り組む人はなかなかうまくいかないかもしれません。というのも、細胞シートの作成と安定した表面を作成するためには、ラングウェアフィルムの分子設計と転写条件を最適化する必要があるからです。
To begin, dissolve styrene, ECT, and ACVA in 40 milliliters of 1,4-dioxane. Freeze the solution in liquid nitrogen under vacuum for 15 to 20 minutes to remove the reactive species. After gradually thawing the solution at room temperature, repeat this freeze pump thaw degassing cycle 3 times. Obtain the polystyrene as a macro raft agent by polymerization at 70 degrees Celsius for 15 hours in an oil bath. Precipitate the polystyrene macro raft agent with 800 milliliters of ether.